You are passing biology by force with biology king Yugi. S6, S5 gather here. In the mock UTEC 2026 item 2, I've not written the whole of it here, but this is the paper of UTEC 2026 mock. Item 2 had 2 tables. The first table, full video will be on YouTube. The first table, table 3, it was about photosynthetic characteristics of moringa trees and cover crops. Table 4, hormonal changes associated with germination of moringa seeds. Now what happened? The item starts by saying that Uganda National Drug Authority wants to increase the production of medicine using natural trees. And the tree that was chosen was a moringa tree that has got some medicinal values. And in that tree, what they wanted is they planted a plantation and they got a manager to look after it, who is an agricultural specialist. And it is after photosynthesis of these trees, the sugars they make are the ones that have to be used to make medicine. You all know that Uganda these days produces a lot of medicines, and most of the sources are natural sources. So the sugars made by moringa trees during photosynthesis were the ones to be used to make medicines. Now the plantation was grown. However, in the plantation of moringa trees, then they put there a cover crop with an intention to prevent weed growth, but also to capture moisture. The plantation was put on a lowland, not highland. Now what happened? A biochemical study between moringa trees and the cover crop. Remember, the intended crop is moringa tree. The cover crop is just to prevent weed growth in the moringa tree plantation, but also to capture moisture in the soil. Now, a biochemical study showed this. Moringa trees, rubisco activity was 58%. However, the enzyme PEP carboxylase was absent. Whenever a student of Form 6 or Form 5 hears that, what does it mean? If PEP carboxylase is absent in the moringa, what does it mean? Answer me. It means moringa is a C3 plant. Now, soluble sugars produced by the moringa were 20 grams per plant per day. But now, to their dismay, the cover crop, which is not the intended crop, it had rubisco enzyme activity 36%, but also PEP carboxylase activity 89%, meaning it had both enzymes, rubisco and PEP carboxylase. Now, soluble sugar produced gram per plant per day were 34, meaning the cover crop, which is not the intended crop, was performing better than the moringa, which is the intended crop here. Now, that one, what does it mean? It means that the cover crop is a C4 because it has got both rubisco and PEP carboxylase. Now, table 2, it shows you germination of moringa, hormonal changes associated with germination of moringa seeds, abscisic acid, gibberellins, amylase, germinating seeds. You are shown values here. You must be knowing as a candidate, recalling that abscisic acid, this is a substance that opposes germination. In other words, it promotes seed dormancy. The higher its concentration, the lower the rate of the higher the chances of seed dormancy. The lower its concentration in the seeds, the higher the chances of germination. Gibberellins promote seed germination. Amylase enzyme catalyzes hydrolysis of starch within seeds for the proper growth of the embryo, because the embryo has to respire to provide air to get energy that will facilitate cell division, so that the shoot and the radicle are formed. Then germinating seeds are also shown here in the first storage condition, cool, moist, and hot, dry. Now, task A, analyze the causes of the differences in sugar production and seed germination. Analyze the causes of the differences in sugar production and seed germination. The first marker given here, you have to quote the trend. Of course, what is going to help us here? In the first place, because here we have two things. Analyze the causes of the differences in sugar production and seed germination. Now, we need to first of all handle sugar production alone. The first marker awarded here is to quote the trend using table 1 where it is sugar production. For example, you say, first quote the difference. So the difference is, the difference is moringa trees are less photosynthetic. Moringa trees produce less sugars. In brackets 20 grams per plant per day, okay, than the cover crop, which produce more sugars, in brackets 34 grams per plant per day. Okay, the moment you cite that kind of difference, very well, you earn yourself an A1 mark. Now we proceed. After quoting the difference that moringa produces low sugars, cover crop produces a high amount of sugars, okay, while quoting also the data from the table, then you proceed. After full stop, proceed and explain. This is because moringa being a C3 plant, due to absence of the enzyme pep carboxylase, but due to presence of rubisco, it is less photosynthetically effective since it is affected by photorespiration, thereby lowering its photosynthetic efficiency. It is affected by photorespiration, lowering its photosynthetic efficiency, hence producing low amounts of sugars. Full stop. Now, the cover crop. Now, if you ended there, someone marking you, and when someone marking you is someone who is very kind, is going to award you an A. You have explained, meaning for it, it's as if you're meaning that the cover crop for it is not affected by photorespiration. But now, if someone wants to win, you continue and also give the other side of the cover crop. The cover crop is more photosynthetically efficient since it is a C4 plant due to presence of both rubisco and pep carboxylase. Rubisco and pep carboxylase. So if you talk about both of them, you'll get yourself an AE. But now another student can go deeper and win themselves an AP, a deep explanation. You can say this is because moringa trees are C3 plants due to absence of pep carboxylase but presence of rubisco. Full stop. Rubisco fixes, has got an affinity of oxygen, and on fixing it, it forms less glycerate phosphate and phosphoglycolic acid, thereby lowering the photosynthetic efficiency since glycerate phosphate is the only one committed to the Calvin cycle to produce sugars. Okay, so another one can say the cover crop is more photosynthetically effective due to presence of pep carboxylase and rubisco, hence fixing carbon dioxide twice. Full stop. Pep carboxylase has zero affinity for oxygen and has higher affinity for carbon dioxide, thereby fixing carbon dioxide into phosphoenolpyruvate to form oxaloacetate. Which is reduced by reduced NADP to malic acid that is shunted from the mesophyll cell chloroplasts into the bundle sheath cell chloroplasts, where malic acid is decarboxylated to release free carbon dioxide and liberate PEP, which diffuses back through plasmodesmata to the mesophyll cells to refix carbon dioxide. Then carbon dioxide is fixed by Rubisco into the Calvin cycle to produce sugars, thereby shielding Rubisco from accessing carbon dioxide and hence promoting higher photosynthetic efficiency. Such a student will also earn themselves an APS, for example, if you use this kind of coding. So members, if the deeper explanation you give in line with what is asked by the question, by the task itself, the deeper the explanation you give, the more the marks. You explain, give examples where necessary. Here we don't have examples that are necessary here. Go deeper. The deeper, the more you shift towards a deeper marking point. You see, AE, AP, and APS, all of them are awarded due to explanations given, but due to they differ in terms of depth. AE is less than AP, AP is less than APS in terms of strength, in terms of scores. So the deeper explanation you give, the more marks you earn yourself. So that is how you had to respond about to task A in line with sugar production. Full stop. Now, when you go to seed germination, this is what we are expecting from you. When you look at seed germination, we have two conditions. Condition one, cool moisty. Under cool moisty, in the moringa seeds, under cool moist, butyric acid concentration is 5, gibberellin is 20, amylase activity is 86. You see? Now, seed germination is 91. But during hot dry, butyric acid is 17. That one should tell you that butyric acid is soluble in water, more soluble in water than gibberellic acid. Now, you see, it is 17. Now, what happens when butyric acid is 17 under hot dry, gibberellin is 8. You see, has reduced. When butyric acid increases, gibberellin lowers. When increase of butyric acid, remember this substance promotes seed dormancy. Amylase activity is decreased. Look, drop rapidly from 86% to 38%. Okay, and consequently, seed germination lowers from 91 to 42. From 91 to 42 per 100 seeds. What does it mean? It means decrease of bearings or increase of butyric acid lowers the rate of seed germination. In other words, promotes seed dormancy. However, the increase of gibberellin and lowering of butyric acid, like in the case one, that one results into increase in the rate of seed dormancy. I think that one is understood. That one should be understood very well. Good. Now, under seed germination, I would say that moringa seeds germinate more. Moringa seeds germinate more, or the rate of germination of moringa seeds is high under cool and moist conditions, 91 seedlings, germinating seedlings per 100 seeds, than under hot, dry conditions where the germination of moringa seeds rapidly decreases to 42 seedlings per 100 seeds. Okay? Germinating, full stop. This is because under cool, moist conditions, abscisic acid concentration is low, in brackets 5 micrograms per gram seed, 5 micrograms per gram seed, and hence its effect to prevent germination greatly reduces. Full stop. Gibberellin is high, 20 microgram per gram seed, hence making the chances of seed germination high, since gibberellin promotes seed germination. Okay? Full stop. Go on, say, under hot and dry conditions, the concentration of abscisic acid is high, 17 microgram per gram seed, yet that of gibberellin is low, 8 microgram per gram seed. Full stop. Full stop. Can go and say the increase of abscisic acid inhibits the activity of the enzyme amylase to catalyze hydrolysis of stored starch into soluble sugars that are used for respiration by the embryo for its growth, hence decreasing seed germination. Now, of course, learners around the country will write different answers. I'm just giving you a possible way of responding to these items. Don't be locked up to such kind of a response, but I'm giving you a clue. Now, the deeper explanation you give, the more the scores you are getting. For example, we have said we are implicating the high rate of germination in the first storage condition, cool, moist. We have implicated it to low abscisic acid, meaning the inhibition to amylase activity is also low, and hence amylase enzyme can easily catalyze hydrolysis of stored starch to provide soluble sugars like glucose, which are required for the respiration of the embryo for its growth. Remember, during the growth of the embryo, it divides by mitosis, and mitotic divisions together with proteins, what have you, the ATP in form of energy in form of ATP, and some in other forms is required. We have implicated this slow germination in under hot and dry. We have implicated to very high given abscisic acid concentration 17, which inhibits amylase enzyme activity to 38%. Hence a decreased rate of hydrolysis of starch into soluble sugars, provide the embryo with energy, and hence a decreasing rate of respiration. Now, my candidates, I would like to request you, while responding to these items, write deep biology. Don't give shallow content. The more you give shallow content, the more you're going to gain at AE, the more you may end at A, whatever, you may not reach at AP. Also, don't forget that there are learners working to get an A, some are working to get a B, some are working to get a C, others are going to get a D, others are going to get an E. You see? Now, what creates a difference? A difference is created by the depth of the content given. So there are two things here. How we respond to the item has got two aspects: quantity and quality. Quantity and quality. Now, quality is to do with the biological depth of knowledge you've put on your paper while responding. Don't give shallow responses. Whenever you think about the fact that there are different levels of scoring, some will get E, D, C, B, E, always think about what must be the creator of the difference here. What creates a difference is the depth of the content you write on the paper. Don't be irrelevant. Don't give irrelevant content. Give content that is in line with the item asked, but go somehow deeper. Because that shallow knowledge you may intend to write, everyone can write it. Then what are you going to write on the paper that most people are not able to do it? That is what will make you an outlier in terms of performance. I sign out. Thank you very much.